Melody Lindsay is currently a postdoctoral scientist at the Bigelow Laboratory for Ocean Sciences in East Boothbay, Maine. Oh, let me take this off. Here we go, that's better. She's developing methods to link cell-specific genome data with activities of single microbial cells from low biomass subsurface environments. She graduated with her Ph.D. in 2019 from Montana State University, where her thesis research focused on how hydrogen from water-rock interaction can fuel microbial primary production in Yellowstone hot springs. It was during her PhD work in Dr. Eric Boyd's geomicrobiology lab that she first fell in love with the enigmatic, I can't say that, you know what I'm saying.
Sorry.
You're killing me. The enigmatic microbialites of Great Salt Lake. She was lucky to work on several projects investigating the microbiology of microbialites and how they contribute to the Great Salt Lake food web. She continues to be intrigued by these unique microbially built mineral structures and hopes she can see them in person again soon. And I have to say, Melody, when she sent me her bio, it's called Microbialite. So Microbialite, welcome. Come teach us.
Thank you, Jamie, for that introduction. Okay, so yeah, thank you for having me. I'm gonna share my screen if that sounds okay. Let's see here. You get to see some really ancient microbialites in my desktop background a little bit. Hopefully that looks okay.
Certainly it looks great.
Enough Zoom-inars here. Yeah. All right, so again, thank you so much, Jamie, for that introduction. I'm here to talk to you about the enigmatic microbialites of Great Salt Lake. And so I became, just a little bit about me. Yeah, like Jamie said, I'm a postdoctoral researcher currently at Bigelow Laboratory for Ocean Sciences, but I first became involved with Bonnie and Jamie through Dr. Eric Boyd's lab, and you were introduced to him a little bit at Montana State University. But I graduated in 2019. Haven't been really out west since then, but I hope to see them again one day. Maybe not during not during a pandemic. But yeah, I'm here today to talk to you about the microbialites and give you a little bit of an introduction on them, especially if you've never heard of what a microbialite is, what they are, and what do they do. So just a brief introduction on why I thought microbialites initially were the coolest things on Earth. Microbialites in the geology world represent some of the earliest evidence for life on planet Earth. And so we have in the rock record these ancient kind of layered mounded structures called microbialites, or a different term is stromatolites, that are from the Archean era. So they're about 3.5 billion years old. So these are some of the first evidence that we have for microbial mats on Earth, and they form these very distinctive macro mounded structures. This is just an artist's depiction of what these microbialites or stromatolites might have looked like on Archean Earth 3.5 billion years ago, when the atmosphere was of a different composition in the oceans. And so that's just kind of what they were theorized to look like. But we can take a look at some examples on modern day Earth of microbialites as well. So on modern day Earth, they are relatively rare. They only happen in a couple of places where the circumstances for their formation are just perfect. And so this is one of the more famous locations, and this is in Shark Bay on the western coast of Australia. But another such location for these rare microbialite structures is Great Salt Lake. And this is a picture I took back in, I think, 2015 of Great Salt Lake microbialites. And I just like how it looks compared to the artist's depiction of ancient Earth. And so if we take a closer look at modern day Earth microbialites and look at their laminated structure, we can see that there are similar patterns to what we see in the ancient rock record. So we see these different layers of microbial mats. And if we take a look at the microbialite from Great Salt Lake, this is a picture I took back in 2014, we can see that there are these layers of microbial mat atop this carbonate rock structure. So I'm going to get into that a little bit and give you a little bit of info on how microbialites are formed. And so microbialites are characterized by microbial mats. And so microbial mat forms on, in an aqueous environment. And typically in a microbial mat that results in a microbialite, this will be formed by a process by photosynthesis. And so as photosynthesis takes place in this microbial mat, the microbes will uptake CO2 and increase the pH and drive the precipitation of inorganic carbonate. And that's what lithifies these kind of domed structures. And as the mat grows, it lithifies and it gets buried. You get this concentric formation of these layered dome structures. Another thing that contributes to microbialite formation is also sediment trapping. So these microbial mats are typically, are often very sticky, and as wave action or other physical processes move around sediment and other particles, like ooids in the Great Salt Lake, for example, that sediment can become trapped in these layers of microbial mat and contribute to microbialite formation. So now I want to get into a little bit more specifically on Great Salt Lake microbialites. So this is a map of Great Salt Lake, and we can see here separated into the north arm, this hypersaline, about 30%-ish north arm, and a relatively less saline, as Jamie said earlier, about 15% salinity south arm. But across both of the arms of Great Salt Lake, there are microbialites, which are outlined here in this green dotted pattern. And so microbialites cover about 20% of the bottom of Great Salt Lake, and this represents the largest expanse of microbialites in the world. So despite the rarity across the world, Great Salt Lake has a lot of microbialites. And they're not just cool because of the early Earth aspect and the kind of rock record that they can inform on, but microbialite or periphyton chlorophyll levels are about 9 times that of the lake phytoplankton because they're so concentrated in these mat formations. And it has been estimated that they contribute about 30% of the productivity that free-floating phytoplankton in the lake contribute as well. So I just want to go into a little bit on the schematic of maybe how microbialites in Great Salt Lake have come into the modern era. And this is a schematic published by Mike Vandenberg from Utah Geological Survey in an article he published in 2019. And so this is what the microbialites might have looked like when they formed these layered mounds. And this is under historic conditions, so a higher lake level. And so these microbialites form these domes, but then as the lake level lowered to current day conditions, the water retreated and these domes were left exposed. And so as these domes were exposed, they kind of fell inwards, forming these kind of ring-like, or I think they're called fairy ring-like structures. And that's what we see in modern day Great Salt Lake on the shores, especially of the South Arm. This is a picture taken of microbialites off of the northern coast of Antelope Island. And you can see that they are in these kind of ringed fairy ring structures covered in a microbial mat that's kind of this brown color. So going back to this map, I'm going to tell you a little bit about a study that we published out of Eric Boyd's lab and with Bonnie and Jamie back in 2016, where we looked at the Great Salt Lake microbialites from both the North Arm and the South Arm. So we're not quite sticking to the South Arm, we're going a little bit back to the North Arm, as Jamie said. So I'm going to tell you a little bit more about the species that inhabit these North Arm microbialites. And here we can see they're these white domed structures surrounded by this pink water, and also of the South Arm microbialites, which are these brown colored fairy rings. And of course, this divergence in salinities of the North Arm and the South Arm, and likely what is causing differences in the microbialites that I'm going to tell you about in a second, was of course the causeway, which was constructed in 1959, which cut off river inflow to the North Arm, as many of you will know, from the Bear River, Ogden, Weber River, and the Jordan River, and it caused this great salinity divergence. So the first question we asked is what species can we look at that inhabit and specifically might even build these microbialite structures? And so we took samples, or Bonnie and Jamie took samples from a North Arm microbialite. And if we zoom in on that, it looks like this. This is about a 2 millimeter by 2 millimeter hand sample. We can see it looks kind of salty, salt encrusted. There's not a lot of greenery, especially as compared with a microbialite from the South Arm. So if we take a close look at a microbialite from the South Arm, already just with a visual observance on this 4 millimeter by 4 millimeter sample, we can see that there are, lots of pockets associated with it, and these green spots, which are likely photosynthetic algae colonizing this microbialite, or perhaps also contributing to the formation of these microbialites. And so using next generation molecular biology techniques, we were able to quantify the amount of biomass associated with these two microbialites. And so this is a plot here on the right showing a proxy for biomass, in this case, the copy numbers of genes or DNA associated with each of these samples for both the North Arm and the South Arm. And we have the biomass that is attributed to the 3 domains of life: bacteria, archaea, and eukarya in these different colors. So on first glance, we can see that the South Arm is winning the battle here for amount of biomass. No, not really a surprise, but we've quantified it. The North Arm biomass is not really even visible on the scale, but it is still there. So if we zoom in on this area here of the plot, we can see there is biomass associated with the North Arm microbialites, albeit at a lower level and a different scale axis than the South Arm. So again, using additional next generation DNA sequencing techniques now, we can take a closer look at the microbialite communities of both the North Arm and the South Arm. And so this is a lot of information on the slide about the identities of species associated with microbialites, but I'll try to take you through it, and I'll leave you with an important point about photosynthetic microorganisms. So these 3 plots are showing the percent community composition of different species for both the South Arm and the North Arm microbialites for each of these 3 plots. And each plot represents, again, a separation of the 3 domains of life: archaea, bacteria, and eukarya. And so we can see that there are stark differences between the community composition of the South Arm versus the North Arm microbialites. But what I want to draw your attention to are 2 species, one bacterial species and one eukaryote species. The bacteria species is this Euhalothece cyanobacteria species, which is present in the South Arm, but not present in the North Arm. And the eukaryote species is this Navicula algae species present in large quantities or large percentages in the South Arm, but not in the North Arm. And so these 2 species present in the South Arm, but not the North Arm, are both photosynthetic. And so this lower biomass and a lack of photosynthetic primary producers in the North Arm is likely due to the restraints caused by increased salinity. And we can see that based on this study. That was one study that we were focused on, but we also wanted to know at what point does a shift in salinity begin to affect microbialites, their primary productivity, and perhaps other aspects of Great Salt Lake food web. So far we have looked at 2 salinities. And so we have the South Arm salinity of around 10 to 15%, depending on the time of year and seasons and stuff like that. And then a salinity of 30% for the North Arm. But the question now becomes, is there an ecological tipping point for microbialite associated biomass or the food web that they likely support in Great Salt Lake? So in 2019, we published a study based on lab-based microcosm experiments where we incubated microbialites at 6 different salinities, now ranging from 8 through 30%, in the ones you see here. And so using this experimental approach, we tracked their productivity, changes in microbial species, and then also downstream effects on some secondary consumers that I'll get into in a little bit. So just some quick results from that study in the microbialite response to changing salinities. So in this case, we focused only on South Arm microbialites and these fairy ring structures we see here. I've just shown this picture again for reference. And we used that in our incubation experiments to test these different salinities. This plot is a plot of the amount of carbon uptake by microbes living in a sample of South Arm microbiolite that we incubated at each of these different salinities. And so we can see that at salinities of around 8 to 15%, and native 15% is the native salinity at which we sampled these microbiolites, we have relatively high carbon uptake and therefore primary productivity. However, at salinities at 20% and above, we begin to see this amount of carbon uptake decrease. So this leads us to the conclusion that salinities over 15% cause shifts in primary productivity and community composition, which I did not show that data, but that's in their 2019 study. And Eric Dunham, who's speaking next, might tell you a little bit more about recent studies in primary productivity. So then it also begs the question, what about secondary consumers? And so I'll just give a quick update on something that other people are much more well versed in than me, but I'll give an intro to the next slide. What species are supported by Great Salt Lake microbialites, or this charismatic megafauna? So if we take a look at a microbialite hand sample, we can see not only these layers of microbial mat, but we see little guest stars here. We have this ephedra larva crawling in between the carbonate structure, feeding on the biomass associated with microbialite, and you also have the brine fly or ephedra casing embedded within this microbialite structure. So I'm just going to take you through a rough estimation of rough estimation of a food web, wherein ephedra or brine flies are supported by Great Salt Lake microbialite periphyton. The microbialites also might contribute to the Great Salt Lake phytoplankton as biomass gets sloughed off of these microbialites. And so this is just a picture showing some brine shrimp, who I don't know what the genders are of them, but they are living happily next to this microbialite in a tank that we had in the lab at Montana State University for a couple years. And so lake phytoplankton can support the Artemia species, or the brine shrimp species. And then of course, the brine flies and the brine shrimp support Great Salt Lake bird populations. And the Great Salt Lake supports over 200 species of birds, over 7.5 million birds annually as a part of the Western Hemisphere Shorebird Reserve Network. And so microbialites represent an important source of primary productivity to the lake. They do not exist in a vacuum. And so going back to the experiment we did with different salinities, we also took a look at Artemia responses to changing salinities. So this plot shows the percentage of shrimp hatched in our little microcosm experiments over a period of about 16 weeks at these 6 different salinities. We can see that at salinities of 8 and 10%, a lot of shrimp were able to hatch. They quickly died off, but there was a steady population that remained. But in salinities of around 15 to 20%, we did finally get some shrimp present in our microcosms after 12 weeks. And then at salinities of 25 to 30%, we never saw any shrimp whatsoever. So that indicates that there is a point, a shifting point at which shrimp no longer are able to survive or are no longer able to feed on microbialite biomass. So I just want to leave you with some final thoughts here. Yeah, I was very lucky to be involved with a couple of studies in Eric Boyd's lab at Montana State University and our collaborators, of course, Bonnie and Jamie. But these are some of the final thoughts I have in general on microbialites. And so microbialites of Great Salt Lake are a unique and necessary ecosystem. They're a source of primary production, and they represent the largest expanse of microbialite structures on Earth, on present day Earth. With our lab-based studies, we saw major decreases in primary productivity at salinities greater than 15%, and the species present also shifted, both in the lake, both the South Arm and the North Arm microbialites, and in our salinity incubation experiments. Looking at Artemia, they did not even hatch at greater than 20% salinity, but they were happiest at around 8 to 10% salinity. And so the final thought here is that major anthropogenic events such as the construction of a railroad causeway in the middle of a lake cause major impacts to these structures at a microbial level, which can also impact macroecology of the lake. So I have many people to thank. I first like to thank Boyd Geobiology Lab at Montana State University and my PhD advisor, Dr. Eric Boyd. Had a great time during my PhD, and I miss both Great Salt Lake and Yellowstone and the lab very much. Thanks to all of our collaborators, particularly Bonnie and Jamie at Westminster, and thank you to the funding sources that made this work possible a couple years ago, particularly the Doyle Stevens Scholarship that I received from Friends of Great Salt Lake a couple years ago. So I'm happy to take questions in the question time in about an hour, but if you can think of any others, feel free to email me. I'm currently a postdoctoral researcher at Bigelow Laboratory for Ocean Sciences, so I took a little bit of a turn. I'm currently focused on deep sea subsurface microbiology, but I love thinking about microbialites still, so happy to answer any questions. And thank you all for having me.
Thank you, Melody.